Our next talk will be presented by Dr. Andrea Giovannucci. He's assistant professor of neural engineering at University of North Carolina, Chapel Hill, and head of the neural engineering laboratory there. Before this, Andrea did his PhD in artificial intelligence at Autonoma University of Barcelona, followed by a postdoc at Princeton. He then served as machine learning scientist at the Flatiron Institute, where he helped to develop the analysis software Caiman for calcium imaging. Andrea's lab spans the realms of neural engineering, experimental research, as well as computational research to define how learning and sensing mechanisms interact within the brain. And today he'll be talking to us about new developments in the online analysis of single and two photon calcium imaging. So thank you so much for being with us, Andrea.
Thank you for the very kind introduction. Can you hear me?
Yes.
Awesome. And can you see the screen, right? My share screen?
Yeah.
Okay. I would like, before anything, thank so much the organizers for inviting me. So far, it's been a super exciting workshop. I'm also a big supporter of open source. I dedicated like the last 8 years of my life to deliver products in some sense that are usable by the community and it can be upgraded by the community itself. Today, I'm going to look at a very, very thin slice of my work because since these 15 minutes, there is not much one can say. I would like to try to give you a take-home message. And this take-home message is related to what is the difference between analyzing data from one photon and two photon imaging. Okay. And in particular, with emphasis on online scenarios. This is the outline of my presentation as I just discussed in my introduction. I hesitate to discuss these slides in front of this audience, but very, very briefly, what is the big difference between one photon and two photon imaging? In two photon imaging, you are able to optically section a plane. So you can go deep inside the brain at some depth up to, let's say, 600 microns with two photon imaging. And you can look at what happens there. You can express some fluorescent cells and have a pretty clear picture of what's happening there. When you go with one photon imaging, what you do is you are integrating light that comes from a very large chunk of tissue. And what happens? You pay the price of this because now when you compare these two movies, you can see that the structure is much less resolved and you see a lot of stuff happening there. That it doesn't come just from one pixel, but it comes from a lot of pixels. Okay. But we need to deal with this because the advantage that comes from the cost, the reduced cost of this type of imaging, the fact that it can be easily miniaturized and mounted on the head of animals, it's a huge advantage. And so basically what you are doing here, you are trading off computational difficulty, if you want, with cost and convenience. So I'm going to try to talk about that. Where is this trade-off? In my opinion. Before we go there, I also like to discuss the two types of analysis that one can carry on. One is batch analysis. That means I have a whole movie available to me, and I can analyze it. Okay. And a second scenario in which I'm looking at just one frame at a time. Each frame arrives, and I'm basically updating what I know about the brain, the state of the brain, the activity of the brain. I find this second type of analysis personally very exciting and I think also very relevant for the future. Why do I think this is interesting? Caleb mentioned it before, closed loop experiments, for instance, right? You might want to know what happens at any given time in the brain because you might use this information, like for instance, to stimulate the brain as Tico is doing. You can modulate the sensory environment of the mouse. You can even control neuroprosthetic device in some far-fetched future. But even more simply, if you have big data streams, what if you are recording 24 hours of data? Analyzing can take weeks, right? Analyzing the type of data. And secondly, what about having feedback about what's going on in your experiment? Right? You remember in the past electrophysiology, we're listening to the sound spikes. What do we do now? You can just look at the screen, and when you have 1,000 neurons, there's not much you can say about what's going on. So it would be useful, for instance, to extract in real time the activity of the brain and maybe perform some low dimensional visualization of what's going on in the brain. Okay, let me give you a little bit of an overview of the analysis pipeline for two-photon imaging and how that adapts to one-photon imaging. Roughly speaking, the steps for analyzing this type of fluorescence imaging type pipeline, and you can even put in voltage imaging if you want, is rather stereotypical. First, you have to correct for motion. Okay, basically what you want to achieve that every pixel in your field of view will always represent the same spatial location. The second part is more or less localizing where are the neurons. Okay, and once you have this information, you want to try to denoise your signal. You want to make it cleaner. You want to increase the signal to noise ratio, and you also want to extract spikes at the end. As you know, the relationship between spikes and the fluorescence traces is very nonlinear and difficult to invert. Although Tico showed very nice results when you're trying to basically increase the frame rate. Now, the point I would like to call your attention on is that this is not simply a segmentation problem. Okay? If you try to segment, just average the pixel of this neuron, average the pixel of these neurons, what you will obtain is a signal that is contaminated. If these two neurons will contaminate each other, so your neuroscience results will be invalid in some sense. And also there is some contamination that comes from the background, a little bit. But sorry. Now, if you compare this, with one photon image, it's even much more complex. Where is this background coming? It's very complex, this type of background. In this side, you see like a little bit of a signal goes up and down. Here you see stuff happening everywhere. Also, neurons are not very well defined. Okay? But you need to solve exactly the same problem. Okay, so let's go through the different portion of this pipeline and how they change between one photon and two photon. The first is motion correction. So motion correction, what is generally done is apply a cross-correlation approach. What you're trying to do, you're trying to match two images, one template, that is your ideal field of view, each frame, and be sure that they're aligned. And intuitively, you can understand that if your images are blurred, what happens is that you will have more difficulty to precisely align. So what can you do to improve the situation? Well, we are lucky because with a simple trick of using a bandpass filter, a spatial filter, we can actually sharpen the edges of objects of a specific structure of a specific size. It is of interest to us. So basically, there is a parameter you can set in our software Cayman called G-sig fit, but the basic idea is that you change the parameter of a bandpass filter, and when you have a much more resolved, if you want, sharp image of your field of view, then just your generic two-photon motion correction algorithm will work out of the box. So we get this out of the way, we can consider that the problem solved. The troubles come when you're trying to extract neurons. So in one of the frameworks that is generally applied for extracting neural activity is what's called the constrained non-negative matrix factorization framework. And very briefly, what you have a movie is represented as the sum of components. Okay, these components have two types of natures. One is a spatial nature, like this one, and some have temporal natures. And also, you have two types of these different couple of components. You have components that represent neurons and components that represent the background signal. In mathematical terms, this corresponds to solve a non-negative matrix factorization problem. I'm not going to get into the detail of this, but here you have the intuition behind that. And why do I want to go into the details of this? Because I think it's key that we understand what this B and F means, because that's what makes the difference, the main difference between one photon and two photon image. The idea is to take this pixel by pixel by frames movie, vectorize it, and then basically having it representing this two-dimensional matrix. Then we can approximate this two-dimensional matrix as the sum, okay, as the product, sorry, of two much smaller matrices. Okay? A, C, that represent neurons, and B and F that represent the background. When you take one of the columns of this vector, we reshape it to the shape to be the original frame, the original field of view. What we obtain actually is the neurons once we solve the optimization problem. and we compute these outer products, we can actually reconstruct the activity of just that neuron into your field of view. You know, that's the key. So this algo, when you solve this optimization problem, what you are doing is really decoupling the effect of each of these neurons on the final move. What happens with the background? Well, it's the same thing. Computationally, the difference is that of having a lot of pixels, right? Of having a lot of pixels that are black, very few pixels will be black. You know, most of them will represent something in the field of view. And equivalently, you know, if you do the same operation as before, you can reconstruct just the effect of the background on your final movie. Pay attention now, these two vectors are small, just one or two vectors. Like while in A, in this matrix, you have a big number of components, which each one for each neuron. Here, normally you have one or two. Okay. So you can, for instance, now remove the effect of the background from your movie, as you can see here on the bottom right image. You can reconstruct your movie using all of them. Okay. Okay. What happens with one photon image? When one photon image is the background is much more complex. This is just a simulation, but gives you the idea. Once you have a very complex background, first it's much more difficult to see neurons, right? And only after doing some processing with very complex, if you want, background models, then we can extract neurons. Because of this big integration of light, you are getting signal from a lot of neurons and structure that are not really in your focal plane. If you try to employ what we used before for one photon imaging, we said just this one matrix that represents the background, what you obtain is what you see here at the bottom right. Okay. So this is representing the movie without the background. So let's say you can remove the background from the movie. If you use a more complex model, which I'm going to introduce shortly, then you are able to very clearly see neuron and you don't have so much contamination. If you use the model that I just presented to you, you see there's a lot of activity that is not explained. So we need something more powerful to explain this better. I said, as an experiment on real data, let's see what happens. You know, I'm running this very complex background model, and then I compare this by not using just maybe one vector to reconstruct it, but 8 and 12. Okay. But still, even when you increase the number of components you used to explain, still that is not enough. So this model is not good to explain what's happening in this big chunk of tissue. So what Peng Cheng did in 2018 is basically using a ring model, an autoregressive model. You're explaining the background as the linear combination of points that are around the neuron. Once you have removed the activity of all the other neurons. So you remove the activity of the neurons from the movie, and now you can represent the background as a linear combination of points around a given dot. What is the problem with this? You know, the number of parameters that you are using is equivalent to the pixel to the square. Okay, many of them are zeros, but your matrix is huge. For a 512 by 512 movie, we're going to get to 68 billion elements. As you understand, so why everybody that is trying to analyze one photon imaging versus two photon imaging comes to me and complain. Why is the memory going so high? Why it's taking 2 days to analyze my data set? It's because this model is particularly complex to deal with. So what recently we came up with is a very clever idea. It's basically, what if we exploit the same idea that is behind convolutional networks, which is called parameter sharing? What if instead of having each of the pixels be represented, you know, by 256,000 parameters, why don't I have a little ring that slides across my field of view, okay, or many little rings, and I just learn them. So I just learned some of these filters, and I use them to represent all my field of view. In this case, just 2 of them. So how many parameters do you have in this case? Just all the elements of these 2 rings are the number of parameters plus the number of pixels for each of them. So you are reducing substantially the number of parameters. And when you reduce the number of parameters, you also reduce the risk of overfitting. When you have so many parameters, it's very difficult to fit to anything. In this case, we have used few parameters, and but we basically slide them across the whole field of view and across many frames, and this prevents overfitting. What you see here is the effect of passing this filtering over the image that I showed you before. Okay, so you will have two of these rings. One of these rings will look like this. Okay, so as you see, it's capturing a lot of the structure, and another one is this one. It's capturing a lot, if you want, of what is the vascular system, etc. Okay, I can explain more of the details in the subsequent discussion. Now, for time, for reason of time, I'm going to show you the results. So these are results when you use a super powerful, the super powerful model I told you about, the autoregressive. It takes a lot of time. Using the ring CNN, you have results that are very, very similar. There is some imperfections here and there, but with much less parameter, you are able to clean up your data very nicely. And we compare these with two other strategies that are used currently for the analysis of Miniscope data, which is high-pass filtering in space the data, or MinPipe, which is using basically an opening, a morphological closure operation to identify neurons. And as you see, the clearest neurons, the clearest shapes are extracted by these two models. This is also quantified in the paper that you can find below. Okay, so where do we go with this? Okay, and what is my laboratory doing in the last 2 years? So what we've been doing is working towards analyzing data at a speed that is compatible with voltage imaging. So TICO, for instance, is capturing data at 500 Hertz. What we want to do is to be able to analyze data sets at this speed. To do so, we exploited accelerated hardware. Recently there's been an explosion in the development of GPUs. So we're going to use that together with the software that is making these GPUs very effective. Software for machine learning, in this case, TensorFlow. So what do? We put one image in the GPU. The GPU will go through a set of operations that I explained to you roughly before, and will spit out this denoised data. This works both for calcium voltage imaging data. What happens? This is just working for 2-photon imaging data. But what I just told you, which is, again, a convolutional, basically, network, can be just put inside here. And that's where we are going to go next, to speed up the analysis of 1-photon imaging and also being able to use it for online experiments. So in summary, that's what I discussed with you today. What is the big problem with 1-photon imaging? In one word, it's the background. Yeah? So that the light coming from some structure that are not of interest in some sense. There are some complex models that are very effective that have been proposed in the past, but they contain a very large number of parameters. And what we are going towards right now is that neural network-based and GPU acceleration, neural network-based model and GPU acceleration, to me, are a promising approach to overcome all this divide, if you want, between the analysis of 1-photon and 2-photon imaging. With this, I would like to thank Eftichios Pneumatikakis and Ioannis Friedrichs. We work together at the Simons Foundation. And this work is, the work of the convolutional networks is mostly with them. Whereas the work on the GPU acceleration is from two very talented graduate students, two very talented students, Changia, a graduate student, and Cynthia, another graduate here at UNC. I will also thank you for your attention, and here you can find all the code if you need it, of course. I hope I closed on time.
Yeah, it's perfect. We have time for one question, if there are any.
I went a bit fast. I'm sorry.
No, that's wonderful.
I'd actually like to ask a question about the background. So one of the things that we see when we look at the raw data is that it often seems like that the background may actually have some signal content in it. Have you thought about posing models such that there's also maybe some some spatiotemporal, like interesting data there in the background?
Yeah. Now that you tell me, I'm thinking about it. But so why do I say so? Because there are tools that, when people talk about mesoscopic imaging, often they refer to the fact that you don't see cells, right? And they have a lot of methods to analyze this data. Like local NMF is one of them. For instance, I never thought about doing this. You clean up the data from your neurons, and now you can use maybe local NMF to actually extract some further information. That's a great idea. I don't know if anybody has ever done that. I think it's a great idea. It's very easy. So basically what you're gonna do, once you estimated your background, right? You can do two things. Once you estimated your neural activity, you just remove it, right? And use the background and use local NMF. One is you directly have your low dimensional representation if you want the background, and maybe you can feed that one to some type of factorization output. Great question.
All right, thanks again, Andrea, for joining us.
Thank you.